Hello dear students, welcome to this lecture series in botany. Today we are going to discuss an important topic, that's phytochrome, discovery, role and mechanism of action. First we will discuss about discovery of phytochrome. The discovery and isolation of phytochrome and demonstration of its importance as a pigment that controls photomorphogenic response is one of the most brilliant and important of all physiological accomplishments. Most of the research leading to phytochrome detection and isolation was accomplished at US Department of Agriculture research station in Beltsville, Maryland between 1945 and 1960. The history of phytochrome discovery was summarized by Harry A. Brothwick. Besides Briggs, Sterling B. Hendricks, another pioneer who described various aspects of the discovery of phytochrome. Brothwick and Hendricks collaborated with experts who were familiar with seed dormancy in many species. They constructed a large spectrograph that with bright light source could separate various colors of light over areas so great that even potted plants could be lined up and exposed to different wavelengths. They obtained an action spectrum with a peak in red wavelengths for promotion of germination of grained rapeseed lettuce seeds, only 50 to 20% of which usually sprout in darkness. It has already been shown in 1930s that red light promoted germination of such seeds, but blue light or far-red light inhibited germination to rates below even those obtained in darkness. Far-red light include the wavelengths just longer than red, covering the range from 700 to 800 nanometers. When the seeds were exposed to far-red light just after a promotive treatment with red light, promotion was nullified. But if red light was given after far-red light, germination was enhanced. By repeatedly alternating brief red and far-red light treatments, they found that the color of last light applied determined whether seeds germinated or not, with red light promoting and far-red light nullifying the promotion. Furthermore, inhibition of flowering in short-day plants by interruption of long night with red light was largely overcome by immediately following red light with far-red light. By that time, investigators realized that a blue pigment was present that absorbed red light, that's called as PR, because its concentration was too low to give color to isolated maize seedlings, in which it was first detected by absorption changes with a spectrophotometer. Researchers also decided that the pigment could be converted by a red light to a form called PFR that absorbed far-red light, a form that eventually proved to be olive green in color, and that the blue pigment could be regenerated with far-red light. The PFR form, that's olive green in color produced by red light, was deduced to be active form, whereas the blue form seemed to be inactive. In early 1960s, H.W. Sagelman and other protein chemists purified phytochromes from homogenates of cereal grain seedlings by using column chromatography and other techniques that are routinely used to purify proteins. They demonstrated that isolated phytochromes change color reversibly upon exposure to either red or far-red light. Phytochrome, a blue protein pigment with a molecular weight of about 250 kilodaltons, was not identified as a unique chemical species until 1959. A key breakthrough in the history of phytochrome was the discovery that the effects of red light on morphogenesis could be reversed by subsequent irradiation with the light of longer wavelength called far-red light. This phenomena was first demonstrated in germinating seeds, but was also observed in relation to stem and leaf growth as well as floral induction. The initial observation was that the germination of lettuce seeds is stimulated by red light and inhibited by far-red light. But the real breakthrough was made many years later when the lettuce seeds were exposed to alternating treatments of red and far-red light. Nearly 100% of seeds that received far-red as a final treatment inhibited strongly their germination rate. Two interpretations of these results were possible. One was that there are two pigments, a red light absorbing pigment and a far-red light absorbing pigment. Second, the two pigments act antagonistically in the regulation of seed germination. Alternatively, there might be a single pigment that can exist in two interconvertible forms, a red light absorbing form PR and a far-red light absorbing form which is called as PFR. Phytochrome can interconvert between PR and PFR forms. In dark grown or isolated plants, phytochrome is present in red light absorbing form, that's called as PR, because it is synthesized in this form only. PFR, which to human eye is blue, is converted by red light to a far red light absorbing form called PFR, which is blue green in color. PFR in turn can be converted back to PR by far red light. This is known as photoreversibility. This reconversion property is the most distinctive property of phytochrome. When PR molecules are exposed to red light, most of them absorb it and are converted to PFR, but some of the PFR molecules also absorb red light and is converted back to PR because both PR and PFR absorb red light. Thus, the proportion of phytochrome in the PFR form after saturating irradiation by red light is only 85%. Similarly, the very small amount of far-red light absorbed by PR makes it impossible to convert PFR entirely to PR by a broad spectrum far-red light. Instead, an equilibrium of 97% PR and 3% PFR is achieved. This equilibrium is termed as photostationary state. In addition to absorbing red light, both forms of phytochrome absorb light in blue region of spectrum. Therefore, phytochrome effects can also be elicited by blue light, which can convert PR to PFR and vice versa. PFR is the physiologically active form of phytochrome. Because phytochrome responses are induced by red light, they could in theory result either from appearance of PFR or disappearance of PR. In most cases studied, a quantitative relationship holds between magnitude of physiological response and amount of PFR that is generated by light. But no such relationship holds between physiological response and loss of PR. Evidence such as this has led to the conclusion that PFR is the physiologically active form of phytochrome. Phytochrome is a dimer that is composed of two polypeptides. Native phytochrome is a soluble protein with a molecular mass of 250 kilodaltons. It occurs as a dimer made up of two equivalent subunits. Each subunit consists of two components, a light absorbing pigment molecule that is called as chromophore and a polypeptide chain called apoprotein. The apoprotein monomer has a molecular mass of about 125 kilodaltons. Together, the apoprotein and chromophore make up the holoprotein. In higher plants, the chromophore of phytochrome is a linear tetrapyrrole that is termed as phytochromobilin. Phytochromobilin is synthesized in plastids. The phytochrome apoprotein alone cannot absorb red or far-red light. Light can be absorbed only when polypeptide is covalently linked with phytochromobilin to form holoprotein. Phytochromobilin is synthesized inside plastids and is derived from 5-amino-levulinic acid through a pathway that branches from chlorophyll biosynthetic pathway. It is thought to leak out of plastids into cytosol by a PASU process. Assembly of phytochrome apoprotein with its chromophore is autocatalytic. Now, characteristics of phytochrome induced whole plant responses. The variety of different phytochrome responses in intact plants is extensive in terms of both the kinds of responses and quantity of light needed to induce the responses. A survey of this will show how diversely the effect of single photo event, that is the absorption of light by PR, are manifested throughout the plant. Such responses can be classified into various types depending on amount and duration of light required and on their action spectra. Morphological responses to the photoactivation of phytochrome may be observed visually after a lag time, that is, the time between stimulation and an observed response. The lag time may be as brief as few minutes or as long as few weeks. The more rapid of these responses are usually reversible movements of organelles or reversible volume changes, such as swelling and shrinking in cells. But even some growth responses are remarkably fast. Phytochrome responses can also be distinguished by amount of light required to induce them. The amount of light is referred to as flux, which is defined as number of photons impinging on unit surface area. The most commonly used units for flux are moles of quanta per meter square. In addition to the flux, some phytochrome responses are sensitive to irradiance or flux rate of light. The units of irradiance in terms of photons are moles of quanta per meter square per second. Each phytochrome response has a characteristic range of light flux over which the magnitude of response is proportional to the flux. These responses fall into three categories based on amount of light required. We have three categories, that is very low flux responses, low flux responses and high irradiance responses. First we will discuss about very low flux responses. Very low flux responses are non-photoreversible. Some phytochrome responses can be initiated by fluxes as low as 0.001 micromole per meter square and they saturate at about 0.05 micromole per meter square. For example, in dark grown oat seedlings, red light can stimulate the growth of coleoptile and inhibit the growth of mesocotyle at such a low fluxes. These remarkable effects of vanishingly low levels of illumination are called as very low flux responses. Now low flux responses. Low flux responses are photoreversible. Another set of phytochrome responses cannot be initiated until the flux reaches 1.0 micromole per meter square and they saturate at 1000 micromole per meter square. These responses are referred to as low flux responses and they include most of the red and far-red photoreversible responses such as promotion of lettuce seed germination and regulation of leaf movements. Both very low flux responses and low flux responses can be induced by brief pulses of light provided that the total amount of light energy adds up to the required flux. The total flux is a function of two factors, the flux rate and irradiation time. Thus, the brief pulse of red light will induce a response provided that the light is sufficiently bright, and conversely very dim light will work if the irradiation time is long enough. This reciprocal relationship between flux rate and time is known as law of reciprocity. High irradiance responses are proportional to irradiance and duration. Phytochrome responses of the third type are termed as high irradiance responses. High irradiance responses require prolonged or continuous exposure to the light of relatively high irradiance and responses proportional to the irradiance within a certain range. The reason that these responses are called as high irradiance responses rather than high flux responses is that they are proportional to irradiance rather than flux. High irradiance responses saturate at much higher fluxes than low flux responses, at least 100 times higher, and are not photoreversible. because neither continuous exposure to dim light nor transient exposure to bright light can induce high irradiance responses. High irradiance responses do not obey the law of reciprocity. Now we will discuss about the role of phytochrome. First we will discuss about circadian rhythms. Various metabolic processes in the plant cycle alternatively through high activity and low activity phases with a regular periodicity of 24 hours. These rhythmic changes are referred to as circadian rhythms. The period of rhythm is the time that elapses between successive peaks or troughs in the cycle, and because the rhythm persists in the absence of external controlling factors, it is considered to be endogenous. The endogenous nature of circadian rhythm suggests that they are governed by internal pacemaker called an oscillator. The endogenous oscillator is coupled to a variety of physiological processes. An important feature of the oscillator is that it is unaffected by temperature, which enables the clock to function normally under wide variety of seasonal and climatic conditions. The clock is said to exhibit temperature compensation. Light is the strong modulator of rhythms in both plants and animals. Although circadian rhythm that persists under controlled lab conditions usually has periods 1 or more hours longer or shorter than 24 hours in nature, their periods tend to be uniformly closer to 24 hours because of synchronizing effects of light at daybreak that's referred to as entrainment. Both red and blue light are effective in entrainment. The red light effect is reversed by far red light, that's indicative of phytochrome. Second is the phytochrome regulates the sleep movements of leaves. The sleep movements of leaves is referred to as nyctinacity. This is the well described example of plant circadian rhythms that's regulated by light. In nyctinacity, leaves or leaflets extend horizontally, that's they open to face the light during day and they fold together vertically at night. Nyctinactic leaf movements are exhibited by many legumes such as mimosa, albizia, samnia, as well as members of oxalis. The change in the leaf or leaflet angle is caused by rhythmic turgor changes in the cells of pulvinus, that is a specialized structure present at the base of petiole. Once initiated, the rhythm of opening and closing persists even in constant darkness, both in whole plants and in isolated leaflets. The phase of rhythm, however, can be shifted by various exogenous signals, including red and blue light. Light also directly affects sleep movement. Blue light stimulates closed leaflets to open, and red light followed by darkness causes open leaflets to close. The leaflets begin to close within 5 minutes after being transferred to the darkness, and closure is complete in 30 minutes. Because the effects of red light can be cancelled by far red light, phytochrome regulates leaflet closure. The physiological mechanism of leaf movement is well understood. It results from turgor changes in the cells that are located on the opposite sides of pulvinus, called as ventral motor cells and dorsal motor cells. These changes in turgor pressure depend on K plus and Cl minus fluxes across the plasma membrane of dorsal and ventral motor cells. Leaflets open when dorsal motor cells accumulate K plus and Cl minus ions, causing them to swell, while ventral motor cells release K plus and Cl minus ions, causing them to shrink. A reversal of this process results in leaflet closure. Leaflet closure is therefore an example of rapid response to phytochrome involving ion flux across the membranes. Now, role of phytochromes in elongation and gravitropism of roots. Gravitropic orientation and elongation of etiolated hypocotyls are both regulated by red light through a phytochrome family of photoreceptors. The importance of phytochrome A and B in these red light responses has been established through the studies of PHY mutants. To identify the role that phytochromes play in gravitropism and elongation of roots, the effects of the red light on root elongation were studied, followed by gravitropic curvature from roots of phytochrome mutants of Arabidopsis, that's PHYA, PHYB, PHYD and PHYAB with wild type. It was found that red light inhibits root elongation approximately 35% in isolated seedlings, and this response was controlled by phytochromes. Roots from dark and light ground double mutants and light grown PHYB seedlings have reduced elongation rate compared with wild type. In addition, roots from these seedlings have reduced gravitropic curvature compared with wild type. These results demonstrate that phytochromes play important role in elongation and gravitropic curvature of roots. Now, role of phytochromes in seed germination. Small seeds frequently require exposure to light in addition to suitable moisture and temperature to trigger their germination. This light requirement serves as a protective mechanism because germination of small seeds far below the soil surface would result in exhaustion of food reserves in the seeds before the seedlings reach soil surface. Early experiments with light requiring seeds involved lettuce seeds that had a low percentage of germination in uninterrupted darkness. But a higher percentage germinated if the seeds were exposed to light. Exposure of moistened lettuce seeds to different colors of light in treatment chambers followed by a return to darkness, it was found that seeds exposed to broad band of red light germinated better than those kept in uninterrupted darkness. Now we will discuss about role of phytochromes in flowering. Many flowering plants use phytochrome to regulate timing of flowering based on length of day and night and to set circadian rhythms, which also play a supporting role in regulating floral transitions, such as entrainment of circadian clocks. Nevertheless, it has long thought that phytochromes are essential for day length measurement in many other plants based on numerous physiological analysis. In short day plants, the PFR form of phytochrome gets converted to PR under slow darkness and far red light. When the PR builds up, floragen gets activated, leading to flowering. In long day plants, the PR form of phytochrome gets converted to PFR under red light. The PFR formed in long day plants results in the activation of floragen and hence flowering. Now, mechanism of action of phytochromes. All phytochrome regulated changes in plants begin with the absorption of light by the pigment. After light absorption, the molecular properties of phytochromes are altered, probably causing the signal transmitting sequences in C-terminus to interact with one or more components of signal transduction pathway that ultimately bring about changes in the growth, development or position of the organ. Some of the signal transmitting motifs appear to interact with multiple signal transduction pathways. Furthermore, it is reasonable to assume that different phytochrome proteins utilize different sets of signal transduction pathways. Molecular and biochemical techniques are helping to unravel the early steps in phytochrome action and signal transduction pathways that lead to physiological or developmental response. The responses fall into two general categories. Number 1, relatively rapid turgor response involving ion fluxes. Second, slower long-term processes associated with photomorphogenesis involving alteration in gene expression. Phytochrome regulates membrane potentials and ion fluxes. Phytochromes can rapidly alter the properties of membrane. We have already seen that the low flux red light is required before dark period to induce the rapid leaflet closure during nictinaste, and that fluxes of K plus and Cl minus ions into and out of dorsal motor cells mediate the response. However, the rapidity of leaflet closure in dark would seem to rule out mechanisms based on gene expression. During phytochrome mediated leaflet closure, the apoplastic pH of dorsal motor cells decreases while the apoplastic pH of ventral motor cells increases. Thus, the plasma membrane proton pump of dorsal motor cells appears to be activated by darkness, and H plus pump of ventral motor cells appears to be deactivated under the same conditions. The reverse pattern of apoplastic pH is observed during leaflet opening. Studies have also been carried out on phytochrome regulation of K plus channels in isolated protoplasts of both dorsal and ventral motor cells from Samnia leaves. When the extracellular K plus concentration raises, K plus enters into protoplasts and depolarizes the membrane potential only if K plus channels are open. When the dorsal and ventral motor cell protoplasts were transferred to constant darkness, the state of K plus channels exhibited a circadian rhythmicity during a 21-hour incubation period, and the two cell types varied reciprocally. That's when dorsal cell K plus channels were open, ventral cell K plus channels were closed, and vice versa. The circadian rhythm of leaf movement has its origin in circadian rhythm of K plus channel opening. On the basis of evidence thus far, we can conclude that phytochrome brings about leaflet closure by regulating the activities of primary proton pumps and K plus channels of dorsal and ventral motor cells. However, some of the effects of red and infrared light on membrane potential are so rapid that phytochrome may also interact with membrane. Such rapid modulation has been measured in individual cells and has been inferred from effects of red and infrared light on the surface potential of roots and oat coleoptiles, where the lag between production of PFR and onset of measurable potential changes is 4.5 seconds for hyperpolarization. Changes in the bioelectric potential of cells imply changes in the flux of ion across the membrane. These findings led some workers to suggest that membrane-bound phytochrome represents the physiologically active fraction. If phytochrome exerts its effects on membranes from some distance, no matter how small, involvement of second messenger is implied, and calcium is a good candidate. Rapid changes in cytosolic free calcium have been implicated as second messengers in several signal transduction pathways. Phytochrome acts through multiple signal transduction pathways. Using biochemical approaches, researchers have shown that signaling involves several different mechanisms, including G proteins, calcium, and phosphorylation. G proteins and calcium. Well-characterizing signaling often involves G proteins. These protein complexes are normally membrane associated and have 3 subunits and bind GTP or GDP on one of the subunits. The hydrolysis of GTP to GDP is required for regulation of G protein function. Microinjection experiments indicate that phytochrome signaling can occur in single cells and does not require light after activation. At least one G protein may function downstream of phytochrome. After the G protein step, there are at least 2 branching pathways. One of these pathways is gene expression and chloroplast development that require calcium and colmodulin. Another one is anthocyanin synthesis, which is calcium independent. For many years, it has been known that cyclic AMP and cyclic GMP are important intermediates in hormone and light-induced signaling in animals. Now, phosphorylation. The evidence for the potential role of phosphorylation in phytochrome action first came from red light regulation of protein phosphorylation and phosphorylation dependent binding of transcription factors to the promoters of phytochrome regulated genes. Some highly purified preparations of phytochromes were also reported to have kinase activity. Kinases are the enzymes that have capacity to transfer phosphate groups from ATP to amino acids such as serine, tyrosine, either on themselves or on other proteins. Kinases are often found in signal transduction pathways in which the addition or removal of phosphate groups regulate the enzyme activity. Phytochrome is now known to be protein kinase. The evolutionary origin of phytochrome is very ancient, predating the appearance of eukaryotes. Bacterial phytochromes are light-dependent histidine kinases that function as sensor proteins that phosphorylate corresponding response regulator proteins. However, higher plant phytochromes have some homology with these kinase domains. They do not function as histidine kinases, instead they are serine threonine kinases. In addition, the recombinant versions of higher plant and algal phytochromes have been shown to be light and chromophore modulated kinases that phosphorylate themselves as well as other proteins. So friends, with this we conclude today's program. See you next time with another topic. Till then, goodbye.